Polymerase chain reaction. First, the introduction. PCR is a method for in vitro amplification of DNA. It has substantially accelerated the pace of research in many fields of biology, both by reducing the time required to perform routine manipulations of DNA and by making new manipulations possible. The technique is based on the following. DNA can be denatured from a double-strand molecule to a single-strand molecule by heat. When the single-stranded DNA molecules are cooled they anneal to a double strand. The process of two single-strand DNA molecules annealing occurs with high fidelity. An example, waste pairs with T and C with G. The polymerase chain reaction is illustrated in figure 1. We will watch it in the next page. Sequentially, the template DNA is denatured by heating. Specific primers designed for the sequence of interest anneal on the corresponding DNA sequence of the template DNA when the reaction is called. DNA polymerase extends the DNA from 50 to 30, building a mirror image of the template strand. At the end of one cycle, two exact copies of template DNA have been formed. Using this technique, DNA can be amplified exponentially. This allows minute amount of DNA from biological sample to be assayed, making the technique extremely powerful for basic science and clinical research. In essence, PCR is multiple round of primer extension reaction in which the complementary strand of a defined region of a DNA molecule are simultaneously synthesized by a thermostable DNA polymerase. During repeated rounds of this reaction, the number of newly synthesized DNA strands increased exponentially so that after 20 to 30 reaction cycles, the initial template DNA will have been replicated several million-fold. This power to faithfully amplify, along with the low cost and simplicity of the method, have made PCR an indispensable tool. In figure 1, double-strand target DNA by heating turns up to single-strand DNA. Cooling allows primer to anneal to DNA. Sense primer, antisense primer. Complementary DNA molecules are generated by DNA polymerase extending temple from the primers. Then, two double-strand copies of DNA are generated after one cycle. Parameters influencing the yield and specificity of PCR. 1. DNA polymerase. Many transstable DNA polymerases are now commercially available, each with different characteristics of thermal stability, exonuclease activity, processivity, fidelity, and reverse transcriptase activity. Tag DNA polymerase with its heat stability, high optimal temperature, and broad pH tolerance is the most widely used thermostable DNA polymerase. The recommended concentration range for tag DNA polymerase is between 1 and 2.5 units per 100 reaction. Enzyme requirements may vary with respect to individual target templates, primers, and suppliers. Tag DNA polymerase from different suppliers may behave differently because of different formulation, assay condition, and unit definition, especially for non-licensed products. If the enzyme concentration is too high, greater than 4 units per milliliter, non-specific background products may accumulate if it's too low. An efficient amount of desired product is made. 2. PCR buffer. Each thermostable polymerase has unique buffer requirements for optimal activity, most requiring trace CL between 10 millimolar and 50 millimolar at pH 8.3 to 9.0. Stabilizing proteins such as PSA or gelatin and a non-ionic detergent such as Tween 20, NP40, or Triton X100 is also usually required. Concentrated reaction buffers are normally provided by the supplier of the enzyme, but for certain applications, other additives such as formamide or glycerol may be essential for an efficiency reaction. 3. Magnesium concentration. Magnesium concentration has a significant effect on the efficiency and specificity of the PCR reaction, in addition to its direct influence on enzyme activity and fidelity. Magnesium also affects the Tm of the various hybrids that form during cycling, including primer template, template, and primer. It is also important to remember that free nucleotide and any editor present in the reaction influence the effective magnesium concentration. If the magnesium concentration is too low, little or no product may be produced. If it is too high, artificial products may result from non-specific priming to template DNA or from primer dimmer. For each new primer pair, it's generally worthwhile to treat the magnesium concentration in 0.5 millimolar step over the range of 0.5 millimolar to 5 millimolar. For DNTPs, a total DNTP concentration between 80 millimolar and 800 millimolar, 20 millimolar to 200 millimolar HDNTP is recommended for most PCR reactions. It is tempting to believe that more is better, but it should be recognized that 5 microgram of PCR product can be synthesized from only 14 nmoles of DNTP. Concentrations higher than 800 millimolar increase the rate at which tag DNA polymerase mesincorporates nucleotide. That effect can be exploited to generate mutant, but should usually be avoided. 5. Template. In some respects, the quality of template DNA is not as critical for PCR as it's for certain other enzymatic manipulation. Evidence for this can be found in the wide variety of sources of DNA that can be successfully amplified, including fossil DNA, entire bacterial colonies, paraffin-embedded tissue, and single human hairs. In some applications, however, the quality of the DNA template is a very important factor. For example, amplification of long spaces of DNA require that the template be prepared in such a way as to minimize single-strand nicks, and a GC-rich template may require higher or prolonged denatured step and the use of a more thermostable polymerase is 10 power 7-fold excess of primer to template is a good rule of thumb for a strand PCR reaction. Because primer concentration above about 0.5 millimolar can result in a detrimental amount of primer dimer formation. The concentration of template DNA is limited to about 50 fm. For 100 microliter reaction, this concentration represents Pg amount of plasmid DNA, Ng amount of bacterial DNA, and microgram amount of human genomic DNA. Under specific reaction conditions, however, genomic DNA from a single cell can be amplified. 6. Cycle parameter. It is critical that complete strand separation occur during the denatured step. A temperature of 94 to 96 degrees Celsius for 1 minute is usually sufficient, but GC research templates may require a higher temperature due to the high molar excess of primer to template. The optimal annealing temperature is dependent on the rate of cooling from the denatured temperature. It is thus usually several degrees higher than the calculated Tm from the primer template. The extension reaction is fully temperature tolerant, and 72 degrees Celsius is optimal for many thermostable polymerases. The duration of the extension step should be adjusted for the length of the amplified region, with 1 minute recommended for target less than 500 base in length, and 2 to 3 minutes for target greater than 500 base to reduce the chance of mispairing that may result from non-specific hybridization occurring during the addition of reaction component, so-called hot start PCR, in which tagged DNA polymerase is added only after the other reactants are raised to a temperature of 70 to 80 degrees Celsius may be recommended. 7. Primer design and concentration. Primers from 10 to 30 nucleotides in length with a GC composition of 40 to 60% are used in most PCR reactions. Obviously, self-complementary and complementary between primers should be avoided, as should continuous runs of the same nucleotide. Primers corresponding to a region of the template containing a high degree of potential secondary structure should also be avoided. Specificity and the efficiency of the reaction is in many cases improved if the 3 end of each primer terminates in 2 or more G or C residues. Due to the increased likelihood of mispraining, however, it is best to avoid an usually high GC content at the 3 end of the primer. Many applications depend on the use of primer in incorporating special features in their design, either to control specificity or to facilitate subsequent manipulation of the amplified product. These features may include modified nucleotide or non-complementary sequence. Primer concentrations between 0.1 molar and 0.5 millimolar are generally recommended. Higher primer concentration may promote mispairing and accumulation of non-specific product, as well as increase the probability of generating a template independent artifact primer dimmer this site. Products are themselves substrates for PCR and compete with the desired product for enzyme, DNA base, and the primer, resulting in lower yield of the desired fragment. Detection and imaging of PCR product. Nucleic acid don't absorb light in the range of wavelengths to which the human eye is sensitive and so cannot be visualized directly. A number of indirect methods have been employed to direct nucleic acid following electrophoresis, both within gel and on the surface of membrane after their transfer from gels. The most common way of detecting nucleic acids in gels is fluorescent dye. Fluorescent dyes, where sensitivity is not of utmost importance, detection of nucleic acid using fluorescent dyes has traditionally been the method of choice. Its chief features being its simplicity and speed. The dye is in many cases simply added to the gel, and electrophoresis buffer. So staining takes place during electrophoresis. After the separation is complete, the gel is exposed to light of a suitable wavelength. The light emitted by the dye molecule is either waved directly or captured by some sort of image recording device. As in all fluorescent events, the light emitted by the dye is of a longer wavelength than that used to excite the dye. Optical filters can be used to prevent light of the exciting wavelength from entering the image system, thereby improve sensitivity. The dye DNA complex often exhibits enhanced fluorescence relative to the unbound dye, a property of the dye that also leads to gather sensitivity. In recent years, the development of more sensitive fluorescent dyes have been supported by the rising cost associated with handling radio-labeled nucleic acid. There have also been improvements in image device, whereas uh, pho- um, using photographic film and an ultraviolet light box was previously the only way to record the image of fluorescent stain gels. Charge coupled device cameras now offer greater sensitivity than film, and a number of very sensitive, dedicated fluorescent imagers are also available. A serious objection to the use of fluorescent dyes for nucleic acid detection is that they represent a safety hazard and must be handled and disposed of accordingly. It should come as no surprise that molecules exhibiting a high affinity for DNA should have the potential to interfere with DNA function. Most dyes used for detecting nucleic acid are mutageneous and are either suspected or confirmed carcinogens. Because it is simple to use and is sufficiently sensitive for most routine producers. Iridium bromide has long been the standard fluorescent dye for staining nucleic acid in gels. Agarose gel are most conveniently casted, run with buffers already containing the dye at a concentration of 0.5 microgram per milliliter. Iridium bromide is an intercalating dye. The planar molecule fits with high affinity between adjacent base pair in the double helix of dsDNA, but is also binds ssDNA and RNA. If dyes include in the gel and running buffer, the process of the separation can be monitored during electrophoresis by using a handheld UV lamp. But iridium bromide staining gels are most commonly viewed by placing them on an ultraviolet light box. The image can be recorded using either a conventional or a CCD camera. Spectrophotometric quantification of nucleic acid. Due to their molecular structure, nucleic acid absorb light of certain wavelengths. For both DNA and RNA, maximal absorption occurs at 260 nanometers. Over a wide range of concentration, a linear relationship exists between the absorption of light and the nucleic acid concentration. Inside a spectrophotometer, the sample is exposed to ultraviolet light at 260 nanometers and a photodetector measures the light that passes through the sample. The more light absorbed by the sample, the higher the nucleic acid concentration in the sample. The amount of light absorbed by a nucleic acid is a function of the molar extinction coefficients of the substituent nucleotide base. The Beer-Lambert law relates the amount of light absorbed at a given wavelength to the molar extinction coefficient and the concentration of an absorbing molecule, A equals Cl, where A is absorbance, E is the molar extinction coefficient, C is the concentration of the solute, and L is the length of the light path in solution measured in centimeters, usually 1 centimeter. By entering the molar extinction coefficients in m power negative 1 times centimeter power negative 1, concentration is obtained in mole per liter. Alternatively, by entering the specific observation coefficient in microgram by milliliter power negative 1 times centimeter power negative 1. Concentration is expressed in microgram per milliliter. Assuming the covent has a base length of 1 centimeter, different nucleic acids are characterized by the following specific observation coefficients: ssDNA 0.027 microgram per milliliter power negative 1 times centimeter negative 1, dsDNA 0.020 microgram per milliliter power negative 1 times centimeter power negative 1, ssRNA 0.025 microgram per milliliter power negative 1 times centimeter power negative 1. Bring this value into the Beer-Lambert equation. An observance at 260 nanometer of 1.0 corresponding to ssDNA 37 microgram per milliliter, dsDNA 50 microgram per milliliter, ssRNA 50 microgram per milliliter. For ssDNA, this conversion factor applies only to longer nucleic acid molecule. Oligonucleotides, an absorbance of 1.0 corresponds to a concentration of 20 to 33 microgram per milliliter, depending on the oligonucleotide length and base composition. The following formula provides an estimate of the concentration where n equal length in base. C b mole per microliter equal wavelength A 2060 divided 10 times n. A more accurate formula accounts for base composition as well as length. C p mole per microliter equal wavelength A 260 divided 100 per 1.5 time Na plus 0.71 Nc plus 1.2 Ng plus 0.84 Nt, where an equal number of each of the four bases A, C, G, and T. On materials, the protocol described illustrates the basic principles and techniques of PCR and can be modified to suit other practical applications. The example chosen use HIV primer pair SK100-450 and SK431 applied biosystem in conjugation with applied biosystem gene AMP, 10x PCR buffer, magnesium dichloride solution, gene AMP, DNTBS, and PCR carryover prevention kit to amplify a 142 base pair DNA fragment from the conserved gag region of HIV using the Amplitech Gold Hot Start process. 1. 10x PCR buffer 100 mm trace KCl 500 millimolar KCl pH 8.3 at room temperature. 2. 25 millimolar MgCl2 solution. 3. DNTBS 10 millimolar stock of each of DATB, DCTB, DGTB, 20 millimolar stock of DUTB, all neutralized to pH 7.0 with NOH. 4. Primer 1 SK145 25 millimolar in 10 millimolar stress HCl pH 8.3 at room temperature. Primer 2 SK431 2K25 25 millimolar in 10 millimolar stress HCl pH 8.3 at room temperature. AMP Earth uracil in glycolysis 1.0 U per milliliter pH 8.3 at room temperature in 150 millimolar NaCl 30 millimolar trace HCl pH 7.5 at room temperature 10 millimolar ethylene dimethyl acetic acid EDTA 1.0 millimolar dispirol DTT 0.05% 20 5% glycerol 7 HIV positive control DNA 10 power 3 copy per milliliter in 10 mg per milliliter human plasmid DNA. Imply tag gold 5 U per milliliter 9 0.5 milliliter microcentrifuge tube 10 thermal cycle PE applied biosystem gene AMP PCR system. Methods 1. Hot start process. In the Amplitech Gold Hot Start process, a master mix is prepared at room temperature, liquefied into individual tubes, and thermal cycled. Assemble the reagent mix as shown here. Reagent value, final concentration. 2. Add 100 microliter of the above reagent mix to the bottom of each gene AMP PCR reaction tube. Avoid splashing liquid onto the tube wells. If any liquid is present on the tube wells, spin the tube briefly in a microcentrifuge. 3. Amplify the PCR amplifications within a programmed thermal cycle. From the Breaking-Elmer DNA thermal cycle 9600 program and run the following linked file. Even though the PCR process has greatly enhanced scientific studies, a variety of problems with the process, easily revealed by sodium bromide stain agarose jet electrophoresis, can and may need to be considered when encountered. For example, unexpected molecular weight size bands, non-specific banding, or smear can be produced. These unexpected products accumulate from the enzymatic extension of primers that anneal the two non-specific target site. Second, dimer can be produced. Primer dimer can arise during PCR amplification when the DNA template is filled out of the reaction. Too many amplification cycles are used, or the primers are designed with partial complementary at their three ends. Note, an increase in primer dimer formation will decrease the production of the desired product. Third, tag DNA polymerase, which lacks the 3 to 5 exonucleases preferring activity, will occasionally incorporate the wrong base during PCR extension. The sequence of tag mesion incorporation usually have little effect, but should be considered during PCR cloning and subsequent cycle sequencing. PCR amplification for user-selected templates and primers are considered fill user when 1. No product bands are absorbed 2. The PCR product band is multi-banded or 3. The PCR product is smeared with fill user can be investigated and turned into successful PCR by manipulation of a number of variables such as enzyme and salt concentrations, denaturation and anneal per extend time and temperature primer design, and not start reducer. When no desired PCR product band is absorbed, initially verify the enzyme addition and per or concentration per treating the enzyme concentration. Second, the magnesium ion concentration is also critical, so care should be taken not to lower than magnesium ion molarity on addition of reagents. An example, buffers containing EDTA will shut out the magnesium ion. The denature and anneal extend time and temperatures may be too high or too low, causing failures and can be varied to increase reaction specificity. Finally, the chemical integrity of the primers should be considered. In case where the PCR product band is multi-banded, consider raising the anneal temperature in increment of 2 degrees Celsius and per hour review the primer design and composition. If a smear of the PCR product band is seen on an acidum bromide stain agarogel, consider the following options initially, individually, or in combination: decreasing the enzyme concentration, lowering the magnesium ion concentration, lengthening and bare or raising the denature time and temperature, shortening the extension time, reducing overall cycle number, and decreasing the possibility of carryover contamination. Finally, in PCR amplifications, where the PCR product band was initially observed and on later trails, a partial or complete loss of the product band is observed, consider testing new liquids of reagent and decreasing the possibility of carryover contamination. For PCR amplifications, using a modified DNA polymerase such as amplified gold, poor product amplification can occur owing to indicate activation of the hot start polymerase. Incubation time, temperature, and pH are critical for hot start polymerase activation. Contaminations added with the target whether remains from the sample's source of or artifacts. The sample's preparation can affect the PCR pH. Contaminants may also directly inhibit the polymerase. Hot start polymerase activation being during the pre-PCR activation step and continuous through the PCR cycle's denaturation steps. The temperature and duration of these steps and the total number of PCR cycles should be optimized. Additional PCR cycles may increase specific product yield without increased background in a hot start PCR. Raising the temperature above 95 degrees Celsius for any PCR step may irreversibly denature the polymerase. Reverse transcriptase polymerase chain reaction. Reverse transcriptase polymerase chain reaction (RT-PCR) is used to assess the level of mRNA expression. An important advantage of RT-PCR is that only a small amount of biological sample is necessary to determine the mRNA level. The limitation of this method for determining gene expression is that RT-PCR combined with visualization using sodium bromide is only semi-quantitative due to the fact that the PCR reaction is linear only within a range of cycles. While RT-PCR is not well suited for the quantitative comprehension of mRNA expression, the technique is useful in screening tissue for the expression of a gene. For example, RT-PCR-based assays are used in the clinical setting for the detection of RNA virus, including respiratory syncytial virus and influenza A virus. Real-time PCR. The technology of real-time PCR developed out of the necessity for a quantitative technique to determine gene expression level in small samples. Measurement of mRNA for a particular gene by real-time PCR is not fundamentally different from reverse transcriptase PCR. Both techniques require mRNA initially to be reverse transcribed to a cDNA molecule. The cDNA is then subjected to PCR. The difference lies in the fact that the instrument where the PCR reaction is carried out is both a thermocycle and a detection device. The reaction mix contains a fluorescent probe that intercalates into the DNA during synthesis. At the end of each PCR cycle, the amount of fluorescence is assessed based on an algorithm. A threshold level of fluorescence must be reached for an mRNA to be considered expressed, and this is expressed as a cycle number. mRNA expression is therefore directly related to cycle number. The higher the relative expression, the lower the cycle number where the curve crossed the threshold. For example, in figure 5b. Gene A has a higher expression than gene B because the cycle number where it crossed the threshold is lower. Glossary of real-time PCR terms. Absolute quantification. The absolute quantitation assay is used to quantify unknown samples by interpolating their quantity from a standard curve as in determination of variable copy number. Allelic discrimination assay designed to type for gene variant either differentially labeled pores, one for each variant or a single probe, and melting cover analysis can be used for this purpose. Alternative methods include dsDNA binding dyes can be used in combination with melting curve analysis. Amplicon, the amplified sequence of DNA in the PCR process. Application point, the point of cycle number varies fluorescence signal which correlates with the initial amount of target nucleic acid during the exponential phase of PCR. Anchor and reporter probes, two partnering light cycler probes that hybridize on the target sequence in close proximity. The anchor probe donor emits the fluorescence to exit the report pore acceptor to enhanced FRET. The allelic discrimination assay, it is important that the reporter probe spins the mutation and has a lower Tm than the anchor probe. Baseline, the initial cycles of PCR during which there is little change in fluorescence signal, usually cycles 3 to 15. Line value during PCR, changing reaction condition and environment can influence fluorescence. In general, the level of fluorescence in any one well corresponds to the amount of target present. Fluorescence levels may fluctuate due to change in the reaction made creating a background signal. The background signal is most evident during the initial cycles of PCR prior to significant accumulation to the target implicon. During this early PCR cycle, the background signal in all well is used to determine the baseline fluorescence. Occur the inter-reaction plate. The goal of data analysis is to determine when target amplification is sufficiently above the background signal, facilitating more accurate measurement of fluorescence. A single reference sample used as a base for relative fold increase in expression studies, assuming a contain reaction efficiency. Coefficient of variation CV used as a measure of experimental variation. It is important that a linear value, e.g. copy numbers, is used to calculate the CV but not CT value, which are algorithmic. Intra-assay CV quantifies the amount of error seen within the same assay. In duplicate and the inter-assay CV quantifies the error between separate assay. CT threshold cycle reflects the cycle number at which the fluorescence generated within a reaction occurs the threshold. It is inversely correlated to the algorithm of the initial copy number. The CT value assigned to a particular well thus reflects the point during the reaction at which a significant number of implicants have accumulated, also called crossing point (CB) in light cycle terminology. Derivative curve. This curve is used in TM analysis. It has the temperature in the x-axis and the negative derivative of fluorescence F with respect to temperature T, shown as the F per dt. On the y-axis, the reproducibility of a derivative melting curve is high with a standard deviation of only 0.1 degrees Celsius between runs. DNA binding agent. A molecule that emits fluorescence when bound to dsDNA. The prototype is cyber green. In real-time PCR, the fluorescence essentially increases proportionally to dsDNA implicants concentration. The problem with DNA binding agent is that they bind to all dsDNA products, specific implicant or non-specific products, misprimitive, target and primer dimmer include. For this reason, anneals using DNA binding agent is usually coupled with melting analysis. Dynamic ring, the ring of initial template concentration over which accurate CT values are obtained. If endogenesis control is used for DDCT quantitation method, dynamic ranges of target and control should be comparable in absolute quantitation. Interpolation within this ring is accurate, but extrapolation beyond the dynamic region should be avoided. The larger the dynamic region, the greater the ability to detect samples with high and low copy number in the same run. Efficiency of the reaction. The efficiency of the reaction can be calculated by the following question. E equal 10 power negative 1 per slope minus 1. The efficiency of the PCR should be 90 to 100%, meaning building of the applicant at each cycle. This corresponds to a slope of 3.1 to negative 3.6 in the CT vs log template amount. Standard curve. In order to obtain accurate and reproducible result, reaction should have efficiency as close to 100% as possible. Twofold increase of implicant and at each cycle. And in any case, efficiency should be similar for both target and reference normalizer, calibrator, endogenesis control, internal control. A number of variables can affect the efficiency of the PCR. These factors can include length of the implicant, presence of inhibitors, secondary structure, and the primer design. Although veiled data can be obtained that fall outside of the efficiency range, if it is smaller than 0.90, the quantitative real-time PCR should be further optimized or alternative implications design. Endogenous control. This is an RNA or DNA that is naturally present in each uh, experimental sample by using a invariant endogenous control as an active reference. Quantitation of a messenger RNA mRNA target can be normalized for differences in the amount of total RNA added to each reaction and to correct for sample-to-sample variations in reverse transcriptase PCR efficiency. Exogenous control. This is a characterized RNA or DNA packed into each sample at a known concentration. An exogenous active reference is usually an in vitro construct that can be used as an internal positive control (IPC) to distinguish true target negatives from PCR inhibition. An exogenous reference can also be used to normalize for differences in efficiency of sample extraction or complementary DNA cDNA synthesis by reverse transcriptase, whether or not an active reference is used. It is important to use a passive reference dye, usually ROX, in order to normalize for non-PCR related fluctuation in fluorescence signal. FAM 6-carboxy fluorescence, most commonly used reporter dye as the 5 end of a TAC-MAN probe. Fluorescence resonance energy transfer (FRET), the interaction between the electronic exact states of two dye molecules, the extinction, the, ex- the extensions is transferred from one the donor dye molecule to the other, the acceptor dye molecule. FRET is distance dependent and occurs when the donor and the acceptor dye are in close proximity. Housekeeping genes. Genes that are widely expressed in abundance and are usually used as reference genes for normalization in real-time PCR, with the assumption of constant expression. The current trend is first to check which housekeeping genes are stable for the target cell or tissue and then to use more than one of them in normalization. Hybridization probe, one of the main fluorescence monitoring systems for DNA amplification, light cycler probes are hybridization probes and are not hydrolyzed by Taq polymerase. For this reason, melting curve analysis is possible with hybridization probes. Hydrolysis probe, one of the main fluorescence monitoring systems for DNA amplification, TAC-MAN probes are an example. These kinds of probes are hydrolyzed by the 5' endonucleases activity of Taq polymerase during PCR. Internal positive control IPC and exogenous IPC can be added to a multiplex assay or run on its own to monitor the presence of inhibitors in the template. Most commonly, the IBC is added to the PCR master mix to determine whether inhibitory substances are present in the mix. Alternatively, it can be added at the point of specimen collection or prior to nucleic acid extraction to monitor sample stability and extraction efficiency respectively. LATE, linear after the exponential PCR, a new form of asymmetric PCR which uses primer pairs deliberately designed for use at unequal concentration. Like typical asymmetric PCR, LATE PCR amplification is efficiency due to improved primer design. LATE PCR design begins with an exponential phase in which amplification efficiency is similar to that of symmetric PCR. Once the limiting primer is depleted, the reaction abruptly switches to linear amplification, and the single-strand product is made from many additional thermal cycles. LATE PCR can, can, can constantly generates strong signals because the absence of product strand re-annealing parameters hindered hybridization of the molecular beacon to its target strand and continue accumulation of that strand beyond the cycle at which symmetric reactions typically plateau. By eliminating the exponential. Phase LATE PCR generates less error scar among replicates when used in conjugation with molecular beacons. LATE PCR result in increased signal density and reduced sample variation. These features are particularly useful for real-time PCR instead with single cell. LATE PCR has been used to directly amplify ssDNA for pyrosequencing. Logged nucleic acid LNA per a new generation of sequence-specific probes designed using LNA, a novel nucleic acid analog, which has enhanced hybridization, performance, and biological stability. Log dilution, serial dilutions in powers of 10, 10, 100, 1000, LUX power TM, light open extension primers, created by Invitrogen. LUX power TM primers include a self-quenched fluorogenic primer and a corresponding unlabeled primer. The labeled primer has a short sequence tail of 4 to 6 nucleotides on the 5 end that is complementary to the 3 end of the primer. The resulting hairpin secondary structure provides optimal quenching of the fluorophore when the primer is incorporated into double-strand DNA during PCR. The fluoro the fluorophore is depunched and the signal increases by up to 10-fold. By eliminating the need for a quencher dye, the LUX power TM primers reduce the cost. LUX power TM vs deck min. Melting cover dissociations analysis. Every piece of dsDNA has a melting point Tm, at which temperature 50% of the DNA is single strand. The temperature depends on the length of the DNA. Sequence order GC, content and Watson-Crick pairing. When DNA bending dyes are used, as the fragment is heated, a sudden decrease in fluorescence is detected when Tm is reached due to dissociation of DNA strand and release of the dye. This point is determined from the inflection point of the melting curve or the melting peak of the derivative plot. Meant by derivative plot is the negative first derivative of the melting curve. The same analysis can be performed when hybridization probes are used as they are still intact after PCR. As hydrolysis probes, for example, TACMAN, are cleaved during the PCR reaction, no melting curve analysis possible if they are used. Match between a hydrolyzation probe and the target result in a lower Tm. Melting curve analysis can be used in knowing and unknowing. New mutation analysis, as a new mutation will create an additional peak or change the peak area. Minor group binders (MGBs). These dsDNA binding agents are attached to the 3 ends of the TACMAN probes to increase the Tm value by stabilization of hybridization, and to design shorter probes. Longer probes reduce design flexibility and are less sensitive to mismatch. Discrimination. MGBs also reduce background fluorescence and increase dynamic ring due to increased efficiency of reporter quenching by allowing the use of shorter probes with higher Tm value. MGBs enhances mismatch discrimination in genotyping assays. Reverse transcriptase control (RTC). A quantitative real-time PCR control sample that contains the starting RNA and all other components for one-step reaction but no reverse transcriptase. Any amplification suggested genomic DNA contamination. Molecular beacons. This hairpin probes contains constant of a sequence-specific loop region flanked by two inverted repeats. Reporter and puncture dyes are attached to each end of the molecule and remain in close contact unless sequence-specific bending occurs and reporter emission (FRET) occurs. Monte Carlo effect. Problems with reproducible quantification of flow abundance targets greater than 1,000 copies by qPCR. It is a limitation of PCR amplification from small amount of any complex template due to differentiation differentiations in amplification's efficiency between individual template in n template concentration. The lower the abundance of any template, the less likely its true abundance will be reflected in the amplified product. Multiplexing simultaneous analysis of more than one target, specific quantification of multiple targets that are amplified within a reaction can be performed using a differentially labeled primer or probes. Implicon or probe melting curve analysis allows multiplexing in allelic discrimination if a dsDNA binding dye is used as the detection chemistry. Normalization, a control gene that is expressed at a constant level is used to normalize the gene expression result for variable template amount or template quality. If RNA quantitation can be done accurately, normalization may be done using total RNA amount used in the reaction. The use of multiple housekeeping genes that are most appropriate for the target cell or tissue is the most optimal means for normalization. Nucleic acid sequence-based amplifications NASBA is an isothermal nucleic acid amplification procedure based on target-specific primers and probes, and the coordinate activity of THREE enzyme, AMV, reverse transcriptase, RNase H, and T7 RNA polymerase. NASBA allows direct detection of viral RNA by nucleic acid amplification. No amplification controls NAC amines enzyme control. In mRNA analysis, NAC is a mock reverse transcription containing all the RT-PCR reagent except the reverse transcriptase. If cDNA or genomic DNA is used as a template, a reaction mixture lacking tag polymerase can be included in the assay as NAC. No product should be synthesized in the NTC or NAC. If the absolute fluorescence of the NAC is greater than that of the NTC after PCR, fluorescent contaminants may be present in the sample or in the heating block of the thermal cycler. No template controls NTC minus sample control. NTC includes all of the RT-PCR reagent except the RNA template. No product should be synthesized in the NTC or NAC. If a product is amplified, this induces contamination, fluorescent or PCR products, or presence of genomic DNA in the RNA sample. Normalized amount of target, a unitless number that can be used to compare the relative amount of target in different samples. Nucleic acid target, also called target template, DNA or RNA sequence that is going to be amplified. Passive reference, a dye that provides an internal reference to which the reporter dye signal can be normalized during data analysis. Normalization is necessary to correct for fluctuations from well to well caused by changes in concentration or volume. ROX is the most commonly used passive reference dye. Primer Express software, a primer design algorithm by API. It designs TechMan, primer, and probe set to be used as standard condition of API real-time PCR equipment. Ensure the molecule that absorbs the emission of fluorescent reporter when in close vicinity. Most commonly used quenchers include DAMRA, DABCYL, and the BHQ. R coefficient, correlation coefficient, which is used to analyze a standard curve. 10-fold dilutions bullet against CT value, obtained by linear regeneration analysis, regression analysis. It should be greater than or equal 0.99 for gene quantitation analysis. It takes value between 0 and -1 for negative correlation and between 0 and +1 for positive correlations. R-square coefficient, usually mixed up with R, but this is R squared, also called the coefficient of determination. This coefficient only takes value between 0 and +1. R power 2 is used to assess the fit of the standard curve to the data point plotted. The closer the value to 1, the better the fit. Cycle PCR, a powerful technique for nucleic acid amplification and analysis that is completed in less than half an hour. Samples amplified by rapid cycle PCR are immediately analyzed by melting curve analysis in the same instrument. In the presence of fluorescent hydrolyzation probes, melting curves provide dynamic dot plots for fine sequence analysis, including SNBS. Real-time PCR, the continuous collection of fluorescent signal from polymerase chain reaction throughout cycles. Reference, a passive or active signal used to normalize experimental result into endogenesis and exogenesis controls are examples of active references. Active reference means the signal is generated as a result of PCR amplification. Reference dye used in all reactions to obtain normalized report signal Rn adjusted for well-to-well variations by the analysis software. The most common reference dye is ROX and is usually included in the master mix. Reporter dye, fluorophore, the fluorescent dye used to monitor implicon accumulation. This can be attached to a specific probe or can be D as DNA binding agent. Relative quantitation. A relative quantitation assay is used to analyze changes in gene expression in a given sample relative to another reference sample, such as relative increase or decrease compared to the baseline level in gene expression in response to a treatment or in time. Ribosomal RNA rRNA, commonly used as a normalizer in quantitative real-time RNA, it is not considered ideal due to its expression level, transcription by a different RNA polymerase, and possible imbalance in relative rRNA to mRNA content in different cell types. In normalized reporter signal, the fluorescence emission intensity of the reporter dye divided by the fluorescence emission intensity of the passive reference dye. Rn positive is the Rn value of a reaction containing all components including the template, and Rn negative is the Rn value of an unreacted sample. The Rn negative value can be obtained from the early cycles of a real-time PCR run. This cycle prior to a significant increase in fluorescence or a reaction that doesn't contain any template. ROX 6-carboxy-X-rhodamine, most commonly used passive reference dye for normalization of report signal. Scorpion, another fluorescence detection system, consists of detection probe with the upstream primer with a fluorophore at the 5 end, followed by a complementary stem loop structure also containing the specific probe sequence, quencher dye, and the PCR primer on the 3 end. The structure makes the sequence specific priming, and probing an unimolecular event that creates enough specificity for allelic discrimination assay. Slope, mathematically calculated slope of standard curve, the plot of CT value against the logarithm of tenfold dilution of target nucleic acid. This slope is used for efficiency calculation. Ideal, the slope should be negative 3.3, negative 3.1 to negative 3.6, which corresponds to 100% efficiency, previously 1.0092. Two-fold precisely 2.0092 amplification at each cycle, also called gradient. Standard, a sample of known concentration used to construct a standard curve by running standards of varying concentrations. A standard curve is created from which the quantity of an unknown sample can be calculated. Standard curve obtained by plotting CT value against log transformed concentrations of serial tenfold dilution of the target nucleic acid. Standard curve is obtained for quantitative PCR, and the range of concentrations included should cover the expected unknown concentrations range. Races primers, originally created by Oncor, sonorized primers are similar to molecular beacons. They are self-complementary primers which dissociate through the synapses of the complementary strand and produce fluorescent signals, currently known as amplifier fluorescent detection system by Chemicon. TAMRA 6-carboxy tetramethyl most commonly used quencher at the 3 end of a Tachman probe. Tachman probe, a dual-labeled specific hydrolysis probe designed to bind to a target sequence with a fluorescent reporter dye at one end and a quencher at the other. Threshold, usually 10x, the standard division of Rn for the early PCR cycles. Baseline, the threshold should be set in the region associated with an exponential growth of PCR product. It is the numerical value assigned for each run to calculate the CT value for each amplification. Unknown, a sample containing an unknown quantity of template. This is the sample of interest whose quantity is being determined.